Okay, shall we start?
Sounds good.
Okay, so this we are competing against the impeachment trial, as Chris mentioned. So it's a very tough competition. So this presentation will be recorded. So please turn off your camera if you are not comfortable. And I am honored to introduce Dr. Christopher Fernandez. Chris joined our department in September 2020 last year to work on the USDA project titled Oak Success Under Pine as a research associate. Chris received his PhD from Pennsylvania State University under the supervision of Dr. Roger Coyle. Then Chris worked as a postdoctoral researcher in Dr. Peter Kennedy's lab at the University of Minnesota. Chris is an expert in forest soil ecology, especially on mycorrhizal fungi. He even had an interview at the Science Friday on that topic. He has published numerous articles in high-profile journals, including Ecology Letters, Global Change Biology, and New Phytologist. Chris is going to talk about mycorrhizal fungi and soil carbon cycling today. It's all yours.
Thank you, Aki. Well, first I'd just like to thank the department for inviting me to talk to you guys today. As Aki mentioned, my expertise is in mycorrhizal fungi, so this talk will be kind of focused on mycorrhizal fungi and their effects on carbon cycling forests. Let's see. So the seminar will have 4 parts. I'm just going to briefly introduce what mycorrhizal fungi are and the various aspects of their biology. Then I'm going to get into some research I've done looking at fungal competition and the effects of competition on soil carbon cycling. Then I'll start to talk about mycorrhizal necromass decomposition. And then I'll briefly wrap it up with a few take-home messages. What are mycorrhizal fungi? Mycorrhizal fungi are incredibly important organisms. 90% of plant species globally have some sort of mycorrhizal association. Here we have pictured the three different main types of mycorrhizal fungi. So you have arbuscular mycorrhizal fungi, which are really important in tropical ecosystems, as well as agroecosystems and prairies and grasslands. Then you have ectomycorrhizal fungi, which are pictured here in the middle, which are really dominant in temperate and boreal forests. And then you have ericoid mycorrhizal fungi, which are really prevalent and dominant in any kind of systems where there are a lot of ericaceous shrubs, such as peatlands and tundras and those sorts of things. Most of my work is looking at or examining the role that ectomycorrhizal play in soil biogeochemistry in forests. I'd say 95% of my work is focused in on this particular group of mycorrhizal association. And again, these are really important in temperate and boreal forests. The basis of any mycorrhizal association is that the mycorrhizal fungus colonizes its host plant's fine roots, and it acquires nitrogen and phosphorus primarily found in the soil and exchanges those nutrients to the host for photosynthate or carbon. So in most natural systems, much of this nitrogen and phosphorus is tied up in forms that are unavailable for direct plant uptake. So this makes these symbioses incredibly important for plant nutrition in these ecosystems and governs productivity in these systems. So there are 3 different main structures of an ectomycorrhizal fungus. And these are particular to ectomycorrhizal fungi, so they don't really apply to other things like arbuscular mycorrhizal fungi or ericoid. But there are three different structures of ectomycorrhizal fungi primarily. First, you have the ectomycorrhiza itself. And I'm going to try to use a laser thing pointer here. So here we have a root with the ectomycorrhizas. And these are essentially just the exchange sites of carbon and nutrients, right? So this is the fungus enveloping the fine root. This is where carbon enters the fungus and nitrogen and phosphorus enter the plant. As here are just 9 different types of ectomycorrhizas. These are produced by different species. As you can see, there's quite a bit of variation in terms of morphology and biochemistry across different species of ectomycorrhizal fungus. So each row represents one host plant colonized by different ectomycorrhizal fungi. So, and this structure where the mycelium wraps around the root is called the mantle, right? And the next structure of an ectomycorrhizal fungus is the sporocarp. And these are often, encompass mushrooms that you see in a forest floor. And basically these are the sites of sexual reproduction and spore production, right? So these are structures that are used for reproducing and dispersal for these fungi. And everyone's familiar with mushrooms. There are some species of ectomycorrhizal fungi that produce underground or hypogeous sporocarps, but basically these are used for sexual reproduction and dispersal. And then finally, we have extramatrical mycelium. And this is really the business end of the ectomycorrhizal fungus. This is just hyphae, which are the filamentous structures that fungi produce that explore the soil solution for nutrients to acquire and pass on to its host. And unlike arbuscular mycorrhizal fungi, ectomycorrhizal fungi who have evolved from various saprotrophic lineages or fungi that break down soil organic matter, they have retained a certain degree of extracellular enzyme production capabilities, right? So this extramatrical mycelium explores the soil, it acquires nutrients, it breaks down organic matter, and acquires those nutrients and passes them back up to the host. So this is actually a picture taken from a site that Aki and Andy and I visited for our project that we're doing here at Michigan State. It's a red pine plantation. And what I did was I just went to a little part of the soil and I dug it up. And this is what you'll see, in most forest systems, right? You'll see all this thread-like structures. This is the extramatrical mycelium of ectomycorrhizal fungi. So the sporocarp, is just the tip of the iceberg. And when you go below ground, the fungus is everywhere. And there's quite a bit of biomass to these fungi. So because these fungi are really important in host nutrition, a lot of nitrogen and phosphorus and carbon cycle through this ectomycorrhizal biomass. So they're important mediators of both carbon and nutrient cycles in these forests. And we're beginning to appreciate just how important they are for driving soil carbon storage. Here are just 2 examples of some high-profile papers that came out in the last couple of years that really demonstrate how important these fungi are for driving soil carbon storage. So how do they drive storage of carbon in soil organic matter? Well, there's 2 primary modes that we think are important for these processes. The first is just the activity of ectomycorrhizal fungi and their competition with free-living saprotrophs. These are bacteria and other fungi that are free-living and are decomposers of soil organic matter. So this is one way that they can drive carbon storage in soil organic matter, is just through their activity and competition with saprotrophs, which can slow down carbon cycling. And another way is the turnover of ectomycorrhizal biomass. So carbon is allocated from the host to these ectomycorrhizal fungi. The ectomycorrhizal fungi produce biomass, right? That biomass then turns over and dies and enters the soil carbon cycle. So these are the two primary modes that we think are really important for ectomycorrhizal fungi driving carbon storage in soil organic matter. And both represent areas that I'm really interested in understanding. And much of my research has been focused around up until now. So I'm going to talk, basically about both of these and some of the work I've done. The first section is focused in on these fungal competitive interactions and soil carbon cycling. So, in ecosystems, the amount of soil organic carbon that is stored in soils is primarily dictated by two processes, the first being productivity and the second being decomposition. So any process that alters these two these two main processes will have an influence on how much soil organic carbon exists in that soil. So, the activity, any kind of activity that influences decomposition of soil organic matter will influence the total pool size, right? So, as I mentioned before, ectomycorrhizal fungi, compete for nitrogen primarily, but also phosphorus found in soil organic matter. So ectomycorrhizal fungi need that nitrogen for their own growth, but also for the growth of their hosts, because they're completely dependent pretty much on this carbon that is allocated to them. Whereas saprotrophs also need that nitrogen, but in addition to that nitrogen, that's their carbon source as well. So, we'd like to think of microbes as these kind of passive organisms, but fungi are incredibly territorial and hyper-competitive. Here are just two pictures of fungal competition studies or experiments done in the lab with wood saprotrophs. And as you can see, these are the same species that are pitted against each other in these microcosms. And as you can see that under certain conditions, they'll be very competitive, and other conditions, not so much. But the main take home from the slide is that these are hyper-competitive organisms that are competing for resources found in various soil pools, right? So how does this influence how much carbon is stored in the soil then? So ectomycorrhizal fungi are kind of unique because they have these hosts, right, that feed them carbon. And it has been hypothesized that ectomycorrhizal fungi are able to mine litter and soil organic matter for nitrogen. What do I mean by that? So they're able to actually extract nitrogen from soil organic matter, leaving behind, carbon and increasing, the carbon to nitrogen ratio of soil organic matter and litter. And what that does is, because saprotrophs rely on both the carbon and nitrogen in these pools, that reduces the activity of saprotrophic activity and the decomposition. So you get this decelerated carbon cycling. I'm sorry, these are advancing without me. So in the 70s, Ruth and Peter Gadgill designed the study that excluded mycorrhizal fungi from the soil mesocosms, essentially, by trenching, right? So when you trench a plot of soil, you're basically excluding ectomycorrhizal ingrowth, right? Because you're cutting off this carbon influx into the soil plots. And what they found was that when you did this, you got this increased decomposition rate, right? And they hypothesized this was due to the exclusion of ectomycorrhizal fungi from the soil plots. So since then, there's been a lot of interest, particularly in the last 10 years, about this so-called Gadgill effect. So this decelerated carbon cycling because due to ectomycorrhizal activity we call it the Gadgill effect. But in 2016, 2015, Peter Kennedy and I were interested in understanding kind of the generality of this phenomenon. So we wrote this review, and up until that point, there had only been a dozen or so studies actually explicitly looking at the so-called Gadgill effect. And we summarized, here's a table from that review, and we summarized the effect of ectomycorrhizal fungi on decomposition. And what we found was that it was a mixed bag, right? So you had some studies where there was a negative effect on decomposition, which would be consistent with the Gadgill effect. Others where there was no effect, and in one case there was actually a positive effect of ectomycorrhizal fungi on decomposition. So, we put forth, there's all these different reasons why there could be, differences in the magnitude and direction of the Gadgill effect, and all these external factors that can control the influence of the Gadgill effect. But one thing that really stuck out to us was how influential the dominant host vegetation was on the presence of the presence of the Gadgill effect, right? So what we wanted to do was design a set of experiments that explicitly tried to tease out the influence of the ectomycorrhizal fungal community, as well as the influence of host vegetation on this Gadgill effect. So what we did was we designed a set of experiments at Cedar Creek Ecosystem Science Reserve. This is a little bit north of the Twin Cities in Minnesota. And you have all these different forest types, primarily either dominated by white pine or northern pin oak, and there's also some maple in there too. So what we wanted to do was select plots that were either dominated by northern pin oak up here or white pine. And within these plots, we kind of set up the classic Gadgill trenching experimental design, right? So we had a bunch of plots that were control plots that allowed access to both ectomycorrhizal and saprotrophic fungi to the soil. And then we also had this trenching treatment, right? So we trenched down to 30 centimeters and excluded, for the most part, the ectomycorrhizal fungi from the soil plots, leaving only saprotrophic fungi access to the substrate that we would incubate. So we incubated litter in these plots. Here's just like kind of the chemistry. We had oak, which is a much higher quality litter, meaning that it had more narrow carbon to nitrogen ratios and lower lignin concentrations. And we incubated each host's litter in the sites that they came from. But we also included a reciprocal litter transplant, right? So we added pine to the oak plots and oak to the pine plots, right? So this allowed us to tease out the influence of litter quality as well as fungal community on this effect. So the kind of hypotheses that we're operating under were, one, the Gadgill effect will be dependent on forest type, right? We thought that ectomycorrhizal fungi in the pine site will reduce soil nitrogen availability. They'll suppress litter decay rates and reduce nitrogen content, because this is what we saw in the literature review. Most of the places where you saw a Gadgill effect happened to be in systems that were dominated by trees that were in the Pinaceae. And we also predicted that the low litter quality of the pine, so high carbon to nitrogen ratio and high lignin to nitrogen ratio, will favor ectomycorrhizal growth, because ectomycorrhizal fungi are not dependent on the carbon found in these substrates. So it doesn't matter that the carbon to nitrogen ratio in these substrates is high, because these fungi are targeting nitrogen, right? And B, they'll have more pronounced Gadgill effects compared to oak litter. Okay, so to kind of test these hypotheses, we used high throughput sequencing, Illumina MySeq. So basically we can quantify and characterize the fungal communities using these DNA sequencing technologies. I'm not going to get too much into how those work, but basically these technologies allow us to quantify the relative abundance of these different fungal taxa in soil, as well as the litter that they colonize. We quantified nutrient availability with these PRS probes. We measured ingrowth of ectomycorrhizas, or ectomycorrhizal roots, into the plots to make sure that the trenching treatment is doing its job. And then we quantified mass loss, elemental analyses, and carbon fraction analysis with ANCOM, as well as characterizing communities for the litter that was incubated in these treatments. So, what we found here is just an NMDS plot of the fungal communities found in the soils at both the pine and oak sites. Basically each one of these points represents a fungal community found in a soil core, found at each one of the sites and within each of the treatments. And the farther apart 2 points are, the more dissimilar the community composition is. So as you can see, the fungal communities found in the pine site are very different from those in the oak site, despite being only meters away from each other. And the trenching treatment, which is depicted in orange and gray, so untrenched being the control, and that's going to be consistent throughout this section. So untrenched control is orange, and the trenched treatment is gray. Had a kind of a marginal effect, and we think that's probably because it takes a lot of time for the DNA to turn over found in soil. So you get this relic DNA, and the effect is not immediate, right? But it did have a kind of a marginal effect. So here is, all of the ectomycorrhizal fungi relative abundance here on the y-axis for the oak site and the pine site, and for each the trenched and untrenched treatments. And as you can see, the trenching treatment reduced ectomycorrhizal abundance in the soil significantly, no matter the site. And this is the root ingrowth, ectomycorrhizal root ingrowth. So ectomycorrhiza is growing into the soil. As you can see, again, the trenching treatment did its job, reducing the amount of ectomycorrhizal ingrowth into the soils at both sites. And here we have the ectomycorrhizal root ingrowth that we just saw on the previous slide, related to ectomycorrhizal fungal relative abundance in the plots. And as you can see, the more ectomycorrhizas growing into the soil, the more, the higher relative abundance of those fungi are in the soil. So that's not surprising at all. It should be that way. However, ectomycorrhizal root ingrowth was only negatively correlated to saprotrophic fungal relative abundance in the pine site. And there was no effect of ectomycorrhizal root ingrowth on saprotrophic fungal relative abundance in the oak site, which was interesting to us. So again, this is consistent with the Gadgil effect, right? So the more ectomycorrhizal fungal activity you have in soils, you would expect, if they're competing with saprotrophic fungi, to see a negative influence on their relative abundance. So in terms of nitrogen availability, so here we have nitrogen availability in the soil, and the treatments, again, trenched and untrenched in the sites, pine and oak. We only saw an increase in nitrogen availability in the trench plot in the pine site. So when we excluded ectomycorrhizal fungi from the plots, you'd expect to see increases in nitrogen availability, right? Because these fungi are pulling out nitrogen from the soil. But we only saw this effect in the pine site and not in the oak site, which is, again, very interesting. So when you regress nitrogen availability onto ectomycorrhizal root ingrowth, so this is, again, the ingrowth of ectomycorrhizas into the soil, and the ratio of ectomycorrhizal fungal abundance to saprotrophic fungal abundance, again, you see this negative relationship, but only in the pine site, and no relationship in the oak site. So again, these are consistent with what you would predict if there was a Gadgil effect, but we only found that in the pine site. Okay, so moving on to decomposition of the litter material that we incubated in the soil. So again, a Gadgil effect would equal this increased decomposition rate. So here we have mass remaining on the Y over time on the X, and this is what you'd expect, right? So the untrenched control will have slower decomposition, so it'll have more mass remaining in its litter over time, relative to the trenched, which we exclude ectomycorrhizal fungi. You'd expect this increased decomposition rate, right? So you get lower mass remaining in those litters. So in the oak litter that was incubated in the oak site, we saw no significant difference between the trenching treatments from 4 months on, but we did see this interesting difference at about 4 months, right? So the untrenched, or the trenched, I should say, actually has slower decomposition than the untrenched, which is the opposite of what you'd expect in if the Gadgil effect was there, right? So trenched treatment had slower decomposition, which is interesting. The oak litter that was incubated in the pine site, again, there was no difference between the treatments across the board from 4 months all the way to 12. And in fact, it was kind of trending again in this opposite direction, where the trench treatment actually had slower decomposition, but it wasn't significant. And when you incubated pine litter, right here, pine litter in the pine site, we saw evidence of the Gadgil effect. So especially at 12 months on, right? So the trench treatment had much quicker decomposition rates than those litters incubated in the untrenched treatment. But when you incubated pine litter in the oak site, again, there was no Gadgil effect present, and there was a little bit of priming again, right? So the trench treatment had slower decomposition compared to the untrenched treatment at 6 months. Sorry. So that's interesting because this is consistent with this idea that substrate matters in determining these interactions or the outcomes of these interactions. So are ectomycorrhizal fungi extracting or mining nitrogen from this litter? So here we have litter type and the lignin to nitrogen ratio. And in the oak litter, there is no difference between the two treatments. But in the pine litter, you can see that the lignin to nitrogen ratio is higher in the untrenched treatment compared to the trenched treatment, suggesting that the ectomycorrhizal fungi are pulling nitrogen from these litter types, from this litter type, leaving behind higher lignin to nitrogen ratios in those substrates. So this is largely consistent with what we see in the literature, right? So everywhere we see a negative effect of ectomycorrhizal fungi on decomposition, it is in a system where there is a conifer, particularly conifers in the Pinaceae. So this led us to kind of put together this this figure. So that hypothesizes where you see a Gad-Gill effect, right? So substrate quality appears to be very important in driving these interactions. So if you have substrate quality that is relatively high, you're not going to find a Gad-Gill effect because ectomycorrhizal fungi aren't going to colonize those litters, right? So, and the saprotrophs will outcompete those ectomycorrhizal fungi. So here's just the oak litter at both pine site or oak site and the pine site in the untrenched treatments. And in blue, we have the relative abundance of saprotrophic fungi. And in red, we have the relative abundance of ectomycorrhizal fungi. And as you can see, the colonization of the oak litter by ectomycorrhizal fungi is relatively low throughout the incubation period. On the other hand, the low litter quality, you see higher colonization of those litters by ectomycorrhizal fungi in both sites, particularly in the pine site. You see this, at 12 months, they're in equal proportion, both ectomycorrhizal and saprotrophic fungi. So, the litter quality determines whether or not ectomycorrhizal fungi will colonize those litters. So if you have high substrate quality, you're not going to find a Gad-Gill effect because those substrates favor saprotrophic carbon use efficiency and result in this coupled carbon and nitrogen cycling. So if you have low litter quality, ectomycorrhizal fungi will colonize, but in the oak site we didn't see the Gad-Gill effect. So we hypothesize this may be because of the ectomycorrhizal fungi in those communities are just non-competitive. So there could be spatial separation across the soil profile. Here's a figure from a paper by Bjorn Lindahl and his co-authors, and there's quite a bit of spatial segregation among saprotrophic fungi and mycorrhizal fungi across the soil profile in a lot of ecosystems. So you have saprotrophs dominating kind of this litter layer, and then you get increases in ectomycorrhizal fungi as you go deeper in the soil profile. So it's possible that in some communities that saprotrophs and ectomycorrhizal fungi aren't competing for the same resources. It's also possible that there are these positive interactions with saprotrophs, so priming. So just like roots, ectomycorrhizal fungi exude a bunch of different compounds that could be beneficial to saprotrophic fungi and lead to increases in decomposition or priming. And this is just a figure from Rich Phillips's classic paper, the RAMP framework, and a recent paper actually showing priming by ectomycorrhizal fungi of soil organic matter, so increased decomposition by the presence of ectomycorrhizal fungi. And it could also just be different functional roles of ectomycorrhizal fungi. So ectomycorrhizal fungi are a very diverse group of organisms that may have very different roles in different ecosystems. So in addition to the breakdown of organic matter for nutrients, a lot of ectomycorrhizal fungi will specialize on mineral weathering. So here's a picture of some feldspar, which is has a lot of phosphorus in it, and you can see the ectomycorrhiza etchings on that piece of mineral, right? So they exude organic acids to break down this mineral to access phosphorus, right? And so this would have no effect on litter decomposition per se. So that leaves us to these communities where there are ectomycorrhizal competitors for these resources in soil organic matter. And in our study, the taxon that kept on showing up in our data sets, associated with this reduced decomposition rates was Tomentella. So Tomentella is not the sexiest ectomycorrhizal fungus. We don't know a whole lot about its the species in this genus's biology, so it doesn't form these sporocarps. It's not well studied. They're not well studied. They're kind of these crusty things, but in every case where we saw reduced decomposition rates, it was associated with species in this genus Tomentella, and we don't really understand why, like what are the functional capabilities, but it is very interesting that this particular fungus, which has a global distribution, is found in a lot of soils and a lot of different ecosystems, but we really don't know a whole lot about it. But it does seem to be very important in driving this phenomenon. Okay, so now I'm going to switch gears and talk about some of my work looking at mycorrhizal necromass. So as I mentioned before, a lot of carbon is allocated below ground to ectomycorrhizal fungi in forest ecosystems, right? So there, estimates range from 10 to 30%, but I think a safe kind of number that to put out there is 20%. So about 20% of host trees' carbon that is fixed is allocated below ground to ectomycorrhizal fungi. So here's just a picture of a pine seedling. This is by David Reed. It's one of the godfathers of mycorrhizal ecology. This is a picture of that seedling, and everything in blue is the actual root system of this pine seedling, and everything else, all this mycelia here, is ectomycorrhizal biomass, fungal biomass, colonizing the root system of this pine seedling. So that's a great deal of carbon. So 20% of net primary productivity is a huge number. Fungi, just like plants and animals, are not immortal, right? So they die. The death of this biomass, therefore, represents this very large flux of carbon into soil carbon cycles. So you have carbon that's allocated to ectomycorrhizal fungi, and the biomass of those fungi, that biomass dies or turns over and then enters this necromass pool. So necromass is just dead biomass. And what we're learning is that these necromass inputs are really important for the amount of carbon held in soils. So in the process, it determines how much carbon enters stable soil organic carbon pools or is respired up into the atmosphere is decomposition. So this necromass, the rate of the decomposition of this necromass will have an effect on how much enters soil organic carbon pools and how much will be respired back into the atmosphere. And this understanding what are the factors that influence these decomposition rates is something that has always really interested me. So a lot of the stuff I'll be talking about for the rest of the talk is kind of work that has come out of my dissertation work. So, in 2008 when I started my PhD, we didn't know much about what controlled the decomposition of fungal necromass. Unlike plant litter, what the factors that control plant litter decomposition, it was relatively unknown. So, we know that initial lignin to initial nitrogen concentrations of plant litters largely determines how fast those substrates decompose, right? So the more lignin and less nitrogen you have, the slower the decomposition of those litters would be. So I wanted to kind of take, what we learned from the plant world and apply it to fungal necromass. As I mentioned earlier, there's a lot of diversity in terms of morphological and biochemical traits of ectomycorrhizal fungi, as you can see here. And this diversity is reflected in their biochemistry. So not all fungi produce the same quantities of these different polymers in their biomass. So the first kind of questions that I developed were trying to understand, what are the different chemical traits that influence the decomposition of necromass. So, to do these kinds of studies, unlike, plant litters where you can go out and collect, leaf litter and, incubate it in soils, you can't do that for fungi. So what you do is you isolate different ectomycorrhizal fungi, grow them in pure culture, and then you can scale up and grow biomass in liquid culture, right? So you can take your isolate, place it in liquid media, and grow that until you have enough biomass built up to make your litter bags. So here's just a picture of some fungi that are growing in liquid culture. Here's what the dried up necromass looks like. And this is very time consuming, and it takes a lot of resources to produce these substrates. So, one flask will produce, about 25 milligrams or 250 milligrams of necromass. And then you can take that necromass, put it into litter bags, and then incubate them like you would leaf litter. Okay, here's a picture of that. So one of the prevailing hypotheses, about 10 years ago was that chitin, which is a fungal specific polymer found in the cell walls of, all fungi, was the cause for a reduced decomposability of necromass. So chitin, again, is just kind of this polysaccharide that has this amide group. But it's very similar in structure to many other polysaccharides. It's that is glycosidic bonds that are tie these polymers together. And if chitin was the reason for reduced decomposability of fungal necromass, you would expect the concentration to increase over the course of decomposition, right? As other polymers are preferentially decomposed. And you would expect the initial concentration to be negatively correlated with the overall decomposability of the necromass. And what we found was the opposite. So we had, this first study had 5 isolates of ectomycorrhizal fungi. Here we have the chitin concentration on the Y, and then a very short incubation time of 30 days. So here you can see that in every case, chitin concentrations were actually reduced over the course of decomposition. So, that first hypothesis was not supported. And when you regressed the total decomposition or dry mass loss onto the initial chitin concentration, you actually got a positive relationship between the two. So, while this was, counter to kind of the prevailing idea that chitin is recalcitrant, it made sense to me because, chitin not only has carbon, it's nitrogen rich, and it has hydrolyzable bonds, right? And you don't need heavy duty enzymes, these peroxidases and so forth to break down the bonds and access those resources, right? So you can get away with hydrolytic enzymes to break these polymers down. So that led me to kind of think of other compounds that might be driving differences in decomposition rates in necromass. And the one that really stuck out was melanin. So unlike chitin, which has concentrations only vary 5 to 10% between two species of fungi, melanin concentrations are very, very different, right? So you have things that produce very little melanin to things that 30% of their biomass is melanin. So, the more melanin a fungus has, the darker it looks. So you have things that are white to brown to jet black. And so this is an ectomycorrhizal fungus, Cenococcum geophilum. Here's the ectomycorrhiza. And this fungus produces a lot of melanin, right? And it doesn't do this for fun. These melanins have functional roles in the survival and fitness of these fungi, and it's linked with all kinds of things like mechanical protection, they're antioxidants, antidesiccants, thermoregulation, photoprotection, all kinds of different roles that melanins play in survivability of and fitness of fungi. And these polymers are really complex, kind of irregularly constructed aromatic polymers, right? So similar to lignin in those respects. And so we devised an experiment looking at how melanin influences the decomposability of necromass, and we found that when you regress the proportional dry mass loss or decomposition onto initial melanin concentrations, you have this negative relationship, suggesting that the more melanin you have in your necromass, the slower you decompose. But there was this one outlier that was really interesting, where you had this one isolate that had really low melanin concentrations, but had also the lowest decomposition rate. So when you take into account initial nitrogen concentration, it explains that outlier in the decomposition. So both initial melanin and initial nitrogen concentration seem to be really good predictors of the decomposition rates of fungal necromass. And when you take the ratio, you get and regress the decomposition rate constant onto that ratio, you get similar relationship as you see in plant litters with initial lignin to initial nitrogen. So, these are the two traits that seem to be really important in determining the decomposability of fungal necromass. And just to kind of reinforce our understanding of the role of melanin in the decomposition of necromass, there was this another experiment in that paper where we inhibited melanin production in Cenococcum with this melanin inhibitor tricyclazole. So this targets these reductase steps in the melanin biosynthetic pathway. And it was done in a very small concentration. So, you don't get a complete knockout of melanin. You just get it kind of reduced to less melanin, about 5% or so. So here's the control without the inhibitor, and here's what the biomass looks like when it's inhibited. And what we found was that relative to the control, you got this increase in decomposition when you inhibited melanin, further supporting the role of melanin as a recalcitrant compound in necromass decomposition. And then recently, we kind of applied what we've learned about fungal necromass to this whole ecosystem climate change manipulation in northern Minnesota. This is the spruce experiment. I'm not going to talk too much about the climate change treatments, just more about kind of what we've learned about fungal necromass decomposition. But there's some interesting results regarding the climate change, but I'm just out of time. So in this experiment, we took, again, necromass with different melanin concentrations, from Cenococcum that was really highly melanized and had, 300 milligrams per gram in its biomass, to Suillus, which is very low melanin concentration. And these, have different carbon and nitrogen ratios as well. And incubated them in these open top chambers. And, what we found was not surprising. Again, we found that here's the proportional mass remaining of the 4 different isolates. The important thing to note here is the time. This is the only experiment that has gone past a year. So this is a 2-year incubation. And what as you can see, there's this rapid decomposition within the first couple months, and then it kind of creeps to an asymptote in all the different isolates. And Cenococcum kind of stalls out at around 35% mass loss. So when you take and these are again 2-pool model decay models. And when you take the parameters in these 2-pool decay models and regress them to initial chemistry of the necromass, you get these significant relationships. So this is the decomposition constant. So this is modeling that fast pool, the decay rate of that fast pool. And when you regress the decay rate parameter K onto initial nitrogen content, you have this negative relationship. So the more nitrogen, the faster the decay, initial decay. And then here is the A parameter, or the asymptote parameter. This is where decomposition kind of stalls out. So, where each of the isolates stall out. And as you can see, when you regress the asymptotic parameter onto initial melanin content, you get this positive relationship. So Cenococcum, which had the most melanin, around 30%, guess what? The decomposition stalls out at around 30%, and this corresponds pretty well with all of them. And when you take the chemistry, so you can take the necromass and apply FTIR spectroscopy to determine the different abundances of these different chemicals found in the necromass. And here we have, all these different things that are associated with polysaccharides and aromatics and amides and so forth. The one thing that we're interested in is kind of these carbon double bonds that are really present in aromatic structures right here. And this would correspond with melanin, of course. And when you take the proportional mass remaining after a year or 2 years, doesn't matter, and regress that onto the peak intensity at that point on the spectra, you have this positive relationship, which suggests that most of the mass still remaining after a year or 2 is actually melanin. Okay, so it's been 10 years since a lot of or 8 years since these studies have started coming out, and we've learned a great deal about what controls the decomposition of fungal necromass. However, there are a lot of knowledge gaps in this whole kind of schematic of the carbon cycle in forests, right? So we have very little understanding of how much is allocated below ground to ectomycorrhizal fungi. We have a handful of studies that give us a kind of a ballpark, but we don't have really good understanding of how much is allocated and what are the different factors controlling that allocation, right? So how much carbon just enters the system as ectomycorrhizal fungal biomass. We know very little about how turnover processes happen to ectomycorrhizal fungi. Like, are there differences in persistence times, and what controls the turnover of this biomass? And a huge knowledge gap is what is important to actually follow this carbon that enters the system as necromass into stable soil organic carbon pools. So there are many different gaps that we don't have a firm grasp on, and this is just scratching the surface of our understanding of these whole processes. Okay, so just have a couple take-home messages. So the first one is fungal interactions are really complex and context dependent, and we should be very cautious about applying certain phenomenon to the globe, like the gaggle effect, like it was in not so long ago. And the gaggle effect is likely controlled by both the community composition found in these forests, as well as the host substrate and the host substrate or the litter substrate chemistry, right? So these are very important in determining whether or not there is this effect present. And two, ectomycorrhizal necromass is an important input into forest soil carbon cycles, and not all this necromass is created equally, and the factors that determine community structure will also probably have implications on how fast that carbon is cycled or how slow it's cycled. And we're just scratching the surface of understanding how these different processes contribute to the formation of stable soil organic carbon. And then finally, I think it's really important to appreciate and understand the taxonomical and functional diversity of these different fungi, because it's not a monolith. These fungi have different traits, and these different traits will have different effects on the soil processes. And it's really important that we understand that before we can start applying, or including these processes into terrestrial biosphere models and so forth. So with that, I would like to thank all these fine folks and funders, but particularly I want to thank 3 undergrads that were kind of instrumental in a lot of this work, Aaron Andrews, Michelle Coblins, and Louis Melke. All these undergrads were really important in a lot of field work and processing a lot of these samples, and I couldn't have done a lot without them. And finally, just want to thank you, and I will open it up for questions in the chat function here, if I can find it. So yeah, thank you.
Thank you, Chris. And I think we can get a few questions, and you can just speak up. I think that's the best way.
Oh, okay.
I got the one question. So you had the pure culture of the ectomycorrhizal fungi, and how challenging it was it without doing it without host trees?
So it depends on the species. So some things you cannot culture, or it's incredibly challenging to culture. Whereas other ectomycorrhizal fungi still have some ability to, process really simple carbon, like, the sugars found in substrate. So you are kind of limited to, a certain subset of ectomycorrhizal fungi when you're doing pure culture work. Some of the most dominant taxa are, you can't work with them in pure culture, like Russula, Tomentella is really hard as well. And Amanita is sort of challenging, but yeah, it's a good question.
Yeah, thank you.
So really interesting results there, and nice talk, Christopher. Appreciated that. So I don't know if you're willing to speculate on this or not, but if we were to think about how to manipulate these systems to increase total carbon storage, right? And I know that you said that Tomentella was one of the EMF genera that was associated with reduced decomposition, which would lead to increased soil carbon storage. But if you were somehow able to manipulate the environment to make it more favorable for Tomentella, for example, and you could slow down decomposition, increase carbon storage, but you would probably, as a result of that, also decrease your nitrogen availability to trees, which would decrease above ground productivity. Do you have any sense of if there's some levers that could be pulled on this to increase total carbon storage in ecosystems?
So the one thing that pops out to me is Tomentella is incredibly common. It's found in a lot of different forest ecosystems. It is really sensitive, though, to disturbance. So once you start introducing disturbance, from climate change to cutting trees down, it tends to go down in abundance in those systems. So just maintaining kind of as close to natural as possible is probably important for maintaining the abundance of Tomentella. But yeah, beyond that, we don't know a lot about Tomentella, in its biology, honestly. Sorry.
Okay, thanks. Well, I'll ask a related question. That was probably too big of a question, and asking you to speculate a little bit more than anyone would be comfortable with. But would increases in Tomentella be associated with decreases in above ground productivity?
Yeah. So it is a effective fungal symbiont, and it seems to be important for host nutrition in kind of a set of unrelated experiments that we're doing or have been conducted in B4 Warmed, which is another climate change experiment in Minnesota. We're seeing that the hosts that have high nitrogen leaf content in their content in their leaves tend to have a lot of Tomentella on their root systems. And once you start introducing warming and reduced precipitation through these rainout shelters, you start to see the host not perform as well because of the various soil moisture goes down, and it doesn't have as much carbon to allocate to its symbionts, and you see Tomentella kind of just crash out of those plants. So you don't see them in the communities anymore, and you get these kind of weedier species colonizing those plants. So they definitely do. Or I would venture to guess that they're very important in terms of nitrogen nutrition to their hosts.
Okay, thanks.
Yep.
Yeah, thank you so much. Any other question?
Hey, Chris.
Hey, how's it going?
I couldn't help but maybe think successionally when you showed some of these, oak versus pine work. And for instance, the graph that you showed kind of relating the saprotrophs to the ectomycorrhizal fungi and the negative relationship for pine, and then the flat line for systems. And I'm wondering, or maybe I'm just being silly and there really isn't ectomycorrhizal tree species that are like later in the successional pathway, but I think there are a few. And like, could you see a positive? Like, so you got the negative with the pine, the flat line with the oak, and then
so the oak stands are a little older, but not by many, maybe 10 years older, I think, than the pine set. So they're, all those plots are kind of in similar successional stages.
Okay. But like, I guess I view, this kind of transition naturally from pine forest to oak to maybe something, even further line on some sites where it's possible to take that successional trajectory.
Yeah, it's something, that I've thought about, but I don't think that study or that experiment addressed it because of the similar ages. We try to kind of, by selecting forest stands the way, in that Cedar Creek site, we try to control for, similar edaphic properties and, all kinds of things, and climate, and just look and try to tease out the contribution of the fungal community and substrate quality on the effect. But yeah, it's something that we have, I've been thinking about, as well as, all these other kind of climatic and other external properties controlling these interactions. I'm sure that, substrate and fungal community composition is not the only thing, are not the only things that are controlling the outcomes of their interactions. And a lot of these, other factors are really, really important as well, like succession. For instance, like, the soil profile, the complexity of the soil profile and, a later stage forest is going to, may just kind of make this these interactions a wash, right? So like you have more niche differentiation across the soil profile as you get into these older stands. But yeah, that's not something, they're pretty young forests, I'd say, comparatively.
Yeah. Well, thank you.
Yeah.
Good presentation. I really enjoyed it.
Thanks, Andy.
Thank you so much. And I think we can just give hands to Chris.
A virtual hand.
Yeah. And so the next one is the 23rd. And I hope to see you all.
Thank you so much.
Thank you.
It's nice to meet you.
Thank you very much.
Thanks, Chris.